The last time someone said Terry needs no introduction, they kept on going and said, so let me tell you something about myself. So I was in New York a few days ago, and I had lunch with Jim Simons, whose foundation is funding this institute and this workshop, the program and the workshop. And you probably know that in addition to the one here at Berkeley, there's also one at MIT, Math Institute. And there's something in New York City called the Flatiron Institute, which now is really big. It's like a data science institute. But he was telling me that they have like 3 big programs, and they want a 4th in computational neuroscience. Wow, this is amazing. You know, we've reached the point now where we're a mature field and actually have resources and opportunities. So if anybody here is interested in being the next director, please let me know. Because he asked me if I had any good suggestions, and you know, it's actually up to you, right? What the future of the field. Okay, so if we look back in the history of neuroscience, right, the traditional way that you record from the brain, in fact, going back 100 years, is from the scalp. And it was already known, you know, back in the days of Hans Berger, that the brain has electrical activity that is constantly changing, and that the patterns of activity are related to your state of behavior. If you're resting and you close your eyes, there's a big alpha component, 10 hertz. And if you're actively making decisions, for example, there's activity in the beta range, which is lower in frequency, or higher actually, roughly 14 to 23 hertz. And then in the 60s, it became possible to record from single neurons, and that really changed the whole landscape, because it was possible to ask a question of what does this specific neuron respond to? What visual stimulus does it respond to? What auditory stimulus does it respond to? What motor pattern is it correlated with in the motor system, right? And if you think about those questions, those are all having to do with representation. How do the neurons represent the world? How do they represent the state of your motor system, right? But as Ela pointed out, the vast majority of the brain falls somewhere between the early sensory processing and the motor output. It's really what we used to call back in the early 80s the hidden units, right? They're hidden from, their activity is really not specified by just the constraints from the world, but also by the intrinsic activity within the cortex. And this is something that has been reported, you know, going back to the very first recordings. It was noticed that they called it spontaneous activity or maintained activity. It was just this bubbling background. But it wasn't just a constant bubbling. It was dynamic. It was changing. It was shifting. It was related to the behavioral state, but in some cryptic way that was impossible to actually easily decode. And Ela's talk shows us that there are now techniques that allow us to get to those internal, latent, hidden states. Now, there's another interesting point to be made, which has to do with the fact that we pretty much, if you really look at trying to understand the global patterns of activity in the brain, and you know, EEG is one example, functional magnetic resonance imaging is another example, right? These global states are really critically important for integrating information. And now let's think about the brain. Okay, so just to calibrate, the biggest deep learning network these days has a few 100 million parameters in it, maybe 1 billion. In 1 cubic millimeter of cortex, there's 1 billion synapses, right? So really what we have is something that's really tiny in comparison to the computational capacity of the entire cortex. The entire cortex is hundreds of thousands, could be, filled with hundreds of thousands of the deep learning networks. And now the problem is, how do you connect them together? How do you integrate all that in representational power? And therefore, this is really the message here, which is that there has to be a completely separate system within the cortex, and in fact within the entire brain, whose primary purpose is communication, right? We'll just look at, we have all these desktop computers. It wasn't until the internet connected them up that it became a powerful, organized system of transferring information, right? So it was the communication that really is as important, or maybe more important, than the actual computing that's going on locally. And how do we understand that? How do we approach it? What are the questions and issues, and who's studying it, right? Okay, well, we'll get back to that later. Now let's go back to Huebel and Wiesel, right? So we know that they were one of the first to actually analyze the response properties of these neurons. Now it's true that if you record the response of a neuron in the visual cortex to its optimal stimulus, you get a bunch of spikes. But if you repeat the same stimulus twice, you don't get the same spike pattern. And as a consequence, they always reported the average. It's called the post-stimulus time histogram, PSTH. And you have to average over 10 or more trials to get something that's reproducible. Because the actual spike timing is variable. In fact, sometimes, and this is really shocking, sometimes there's no response. What's going on here? I thought the brain was reliable here, but...
Terry, there's no response behind you.
No. I think the computer wants to go to sleep. Okay. Okay, and I'm going to illustrate that now in recordings from Dan Feldman, who's here at Berkeley. What he did here was to stimulate one whisker in the a rat's pad of whiskers. And there's a mapping from the whisker pad, whisker by whisker, to the cortex. They're called barrels in the cortex. And if you record from the barrel of a whisker who's stimulated, you're stimulating, and recording now from a layer 5B cell in barrel cortex, it means lower part of the, the part of the cortex actually projects out subcortically. So here's the stimulus, and you can see that the PSTH is very nice. It gives you a burst here, and then there's another burst here. But wait a second, let's look at individual trials, right? You look at this trial. There were no spikes, or sometimes just one spike, right? We're not talking here about something that can be characterized by a firing rate for a single trial. By the way, the animal can detect that slight movement of the whisker very, very accurately. So that means it can somehow integrate over all the neurons. But each individual neuron, as you can see, and this is now from different parts of the whole system of the barrel system. So this is really an interesting question because it's related to the spontaneous activity question, and it is related to an observation first made by Miriam Grünwald, who was one of the first to get voltage-sensitive dyes to work in the cortex, and this is in the visual cortex. And so you're looking down, and this is a monkey, and if you stimulate the right eye or the left eye, you get these ocular dominance columns. So this below this is primary visual cortex V1, and you see above that it looks different. That's V2. So there's a border here. So here's the experiment. It's very simple. And the color coding here is the degree of activity as measured by a voltage-sensitive dye. So this is milliseconds after the stimulus. The stimulus consists of a spot of light, a very tiny spot of light. And you're just looking at what it would be if when you're looking down at the cortex. And you can see there's a delay here, and it's 48 milliseconds before you start seeing something. Now the actual spot itself on this is about 4 millimeters across. So the actual spot itself is probably about this size. If you just look at the neurons that are directly connected to that spot from the retina. But you can see that it grows and it grows and it grows over 100, 200 milliseconds, spreading out. Now, you get spikes in the center, but you don't get any spikes in the background. And why is that? And the reason is that voltage-sensitive dyes are giving you information about subthreshold activity. Just what's happening is that these cells are actually around it, are being depolarized, maybe not to threshold. And so this is really evidence that there is a traveling wave that starts out at the point of the stimulus and spreads out over many millimeters of the cortex. We even know the substrate. There are something called long-range horizontal connections, and they tend to be fairly slow, and which may account for the fact that it takes 100 milliseconds or more for that information to spread out. Okay, so this was a mystery at the time. And people, how do you interpret this, and why is it there? Because if all you see is spikes, the picture looks very different. But it clearly, if information is coming in at several different places on the cortex at the same time as it is in natural scenes, then all of these the cells here are going to be getting subthreshold activation from many different parts of the visual field at different time delays, right? So that's really what we want to explore.
Is it count to imaging the data you showed?
Yeah, this is okay. That's right. To explain to you, there's a hole in the skull of the monkey, and a camera is looking down. And they put a dye into the cortex, which gets into the membrane and reports through small changes in the amount of light coming back what the membrane potential is, right? And most of the membrane potential is in the neuropil. It has to do with synaptic activity. Membrane potentials, not spikes, right? So that's what you have to know.
So what scale is this?
Pardon?
What scale is this?
This is about 4 millimeters across.
Oh, so this is a large area.
Yeah, it's huge. It's actually very, very large, right? It's covers several columns here. Okay, now let's jump to the hippocampus. So here's the hippocampus spread out, sort of peeled back. It's kind of wound up like a French roll. But the area that I'm going to be focusing on is CA1, which is the output from the hippocampus. And I want to focus here at the top. It's called the dorsal surface of the top. And it's known that if you record from one of these neurons in a track where the rat or the mouse is running along, that there will be places along the track where their neuron will suddenly start firing. And in the case of dorsal hippocampus, it'll be centimeters to a meter in terms of the spatial extent. And there are place fields all the way along in different parts of this dorsal hippocampus. Now, as you go down, and this is only known very recently because most people put their electrode from the top and they stop when they get here. But if you go down ventrally to the bottom, it turns out the place fields get bigger and bigger and bigger, and to the point where they become so big that they cover, the whole the entire little ring. Normally they call them mazes, and they're typically about a meter or two around. And it wasn't until people realized that if you really want to understand place fields with are called grid cells, you need to go down the hallway, like 10, 15 meters in order to be able to see the regular patterns here of place fields. So this has been known for a long time, and this goes back to John O'Keefe in the 60s, and Nobel Prize was given for this just a couple years ago. Okay, now in addition to these place fields, there's something else that is very dramatic and extremely well characterized that is going on dynamically. So when the rat is actually exploring its environment, there is a regular frequency. It's called the Theta oscillation in the hippocampus, and it's 4 to 10 hertz. And you can see this is actually in the slice. And this is actually now in vivo. So it's not quite as nice in the slice, just to compare. This is really an oscillation. So this is a little bit more ragged. Let me take you through it. So this is a depth electrode that has 16 different pads where you can record activity. And here they're recording the local field potential, which is the sum of the input from a lot of local circuitry. And at the very top, we're in cortex, and the very middle here, we're in the hippocampus, and the top part, the pyramidal cell layer in the middle, and then the bottom part of the hippocampus. And then it actually gets down to another part of the hippocampus called the dentate gyrus, where the inputs are coming in. But what I wanted to show you is that, first of all, the not perfectly regular, but the actually quite periodic and repeatable oscillation in that 4 to 8 hertz range. That's the theta oscillation. And in passing, I just want to mention that if you look at the crest here, you'll see that there's also another oscillation at a higher frequency called the gamma oscillation. And that's on the order of 30 to 80 hertz. And so there's nested oscillations here. It's not like, there's one buzz. It's like, there's several at the same time, and they're nested. And this is dentate. Look at how big the theta is there. So the theta really extends across the whole hippocampus. And it's also found, by the way, in the entorhinal cortex that feeds into the hippocampus. So this is like a very widespread organizing temporal signal. Now, again, this has been known for 50 years, and it's enormous, and it's very regular, and it goes on. It's a real oscillation. But it wasn't until Sejnowski, who's at Caltech, put an array of electrodes across the hippocampus that we actually saw the spatial organization, right? So here it is. This is the long axis of the hippocampus. And here's the array. And let's now look at the activity pattern on the array during a time when the animal is locomoting. So red is when it's at its peak, and blue is when it's at its nadir. And I think it's pretty clear that this is not an oscillation, it's a traveling wave, right? Is everybody convinced?
Right.
So this has been around for 10 years. Nobody knows what to make of it. I mean, the people who do the hippocampus know. It's really funny. You talk to them and say, oh yeah, I remember that paper. But basically you ignore it. Why do people ignore it? They ignore it because they can't fit it into anything. It doesn't make any sense, right? Why should a traveling wave be there, right? What's it doing there? Okay, well, let's take it seriously. Okay, so I've already showed you this on the left. But let's now look at what this traveling wave means for these different parts of the hippocampus, right? So this is that longitudinal axis that you saw in the array. And what it means is that if you look at the local field potential here, it's going to reach peak 70 milliseconds before it reaches peak down here. So there's a temporal gradient. And by the way, the cells actually fire when there's a peak. It's actually at the minimum of the local field potential, but it's at the peak with the membrane potential. But the point is, though, that there's temporal offsets. And this is going to be really, really important, because really the thing that's been ignored universally almost in most of the work that's been done in vision and even in the hippocampus is time delays. Time delays are ubiquitous. I mean, they're axonal time delays, right? So axons they're really lousy wires, right? They transmit information on the order of meters per second. I mean, compared to the speed of light, that's really lousy, for your computer, right? Literally, you could send signals back and forth with near the speed of light. So, is it the case that nature can't do any better? Or maybe because it had these slow axons, it actually decided to take advantage of them, right? Well, let's find out. Let's take a look at what they could be used for. So one of the interesting the issues has to do with when is it that you change the strength of a synapse? And it turns out that here, timing is really important. So this is a classic experiment that was done in the hippocampus by Booming Poo. And it represents, this is in a slice where you are recording simultaneously from two neurons, and you're able to stimulate both of them at different time points, right, relative to each other. And here, focus up here. This is when, over here on the right, when the presynaptic input is activated through neuron A, and you can see that the postsynaptic cell membrane potential is increasing because that's the synaptic input. And then after a slight time delay, which is along this axis, you stimulate the postsynaptic cell, right, with an action potential. And you pair that at some frequency. And you come back, you compare the strength of that synapse before and after the pairing, and if it's within a 20 millisecond window, you can double the strength. That's called long-term potentiation. But if you reverse the order, and now you do post before pre, so here's that synaptic potential, within a 20-minute window, you can decrease the strength. That's long-term depression. So they're like really exceptional precision here, because we're talking about difference between depression and potentiation in the millisecond range. Right? This is really high precision, and this is really incredibly important. It makes a big difference whether you're going to increase or decrease, right? So that's one thing that's important. The other thing is that if you pair this at less than 10 hertz, like 5 hertz, you don't get this. It has to be 10 hertz or higher. Okay, well that's interesting too, because it means that there's several necessary steps here: precise timing, and number 2, high enough frequency.
I'm sorry.
Pardon?
High enough frequency.
Oh, frequency of pairing, where you stimulate pre and post with that time delay. Yeah. Okay, we're going to come back to this now, because this is going to turn out, all these little pieces I've been giving you are like little breadcrumbs that are out in the literature. I mean, literally hundreds of papers all over the place that are giving us some hints. But unfortunately, they're very difficult to put together. It's like a puzzle, right? You have a jigsaw puzzle, and you take a couple of pieces out, and you begin to see the relationships. Maybe the colors are similar here, and maybe the shapes are similar here. But somehow, as you have more pieces of the puzzle, you begin, the picture seems to come out. But we're still at the stage where we have lots of pieces, but not a lot of patches yet put together. And I'm going to try to put together one of these patches for you. Now, sleep has been already mentioned a couple of times, and it's one of the most remarkable behaviors that we do every day, every single one of us. I shouldn't actually presume. How many people here went to sleep last night? Put up your hand. Okay. Somebody there.
How many people wish they went to sleep?
Right.
We're sitting on airplanes.
Well, that's the point, is that if you don't get your sleep, then you have to pay the price, right? Sleep deprivation. And so it's literally a third of your lifetime you're knocked out. And why? And we actually have many, many theories and almost no experimental evidence that really pins it down. And it's very likely it's not for just one thing. There are many, many things that are going on in your brain. But one thing that's really, we know for sure, is that it's not that your brain turns off, right? You think that if you're conserving energy, well, why not just turn it off, right? That would be the sensible thing to do. No. It actually is almost as active as it is during the day, but the patterns of activity have changed dramatically. And in particular, you get in, during the night, very low frequency waves that are oscillations that are not connected to anything in the world, completely intrinsically generated. And it changes during the night. So when you fall asleep, you go through a sequence of stages until you get to slow wave sleep, which is 3-4 down here. And that's when you have the lowest frequencies, 2 to 4 hertz, very high amplitude. And it's spatially and temporally coherent. And that's what's really neat, because it means that there's a global state there. It's not just processing information locally in different parts for different purposes, different sensory inputs and so forth. It's the whole cortex suddenly is somehow been stitched together. And now, during the night, you periodically go between slow wave sleep and rapid eye movement sleep, which you heard about, dream sleep. And back and forth, spending less and less time in slow wave, more and more time in rapid eye movement sleep, until finally you wake up at the end of 8 hours. Now, and here are representative EEG recordings, and so you can see that the amplitude for awake and REM, which is down here, are very small. They're very similar, in fact. The textbook says you can't discriminate them, but in fact, with machine learning, you can. We have. But compare that to the slow wave sleep. Look at these huge 2 to 4 hertz delta waves. Now, the stage that I want to focus on is this intermediate stage 2. And in fact, most people spend about half of their time in stage 2, because you have to go between REM and slow wave. You always stop at stage 2. And what's going on in stage 2? In fact, it's very easy to identify, because that's when sleep spindles occur. Sleep spindles occur thousands of times during the night. They last 1 or 2 seconds. They're on the order of 10 hertz, the same band as the alpha band. And they consist of bursting. We know that they're generated in the thalamus. The thalamus is the relay station that takes input from the sensory periphery into the thalamus, and then from there it relays it up to the cortex. And in addition to the feed forward, there's also connections from the cortex, layer 6, which is the bottom of the cortex, down to the thalamus. So there's actually a loop between the cortex and the thalamus. And we now know that for spindles, and I've spent 20 years studying this with Mircea Steriad, David McCormick, we have very detailed biophysical models down to the ion channels. And I can tell you in exquisite detail how they get started, how the thalamus recruits the cortex, and through the feedback connections become spatially coherent. Right? That's one of the ways that the thalamus is able to organize the cortex. Okay, so that's what we've been studying, and typically studied studied in the CAD. This is by Mircea Steriad, by putting a bunch of electrodes in, say, the thalamus, and then only 2 here in the cortex, but at a distant location. And here's the recording over about a minute. And you can see that they recur every 5 or 10 seconds. And here's 2 spindles that are blown up. And it's pretty convincing that, they really do are both, widespread, and also it starts a little bit first in the thalamus, but then recruits the cortex. And with the existing techniques that they had, it looked like they were pretty much synchronous. And this was something that David and Mircea and I often debated. What do you mean by synchronous? Do you mean that it's within, it's a 100 millisecond period? So is it okay to be within 10 milliseconds of each other? 5, 20? What's the definition of synchronous at this point, if you're talking about the whole cortex, right? And in some ways, part of the problem was that people never put more than 1 or 2 in the cortex at the same time. To do it properly, you'd have to have a whole array to actually be able to make measurements across the whole cortex. Okay, well now let's think about the one possible function for which there's actually increasing evidence, which is that we know that from many, many psychological experiments that if you do some learning and then take a nap and then come back, you're able to do the task better, whether it's remembering things that you've associated, or even perceptual learning, being able to detect weak stimuli, or motor learning, right? So it's thought from psychologists that there must be something going on in your brain during sleep which helps you consolidate that experience that you just had. And we actually now have a theory based on recordings from rats that actually buttresses that theory. And so here's how the theory goes. This is coming from Yuri Buzaki, who's one of the leaders in recording from the neurons, different parts of the hippocampus and the cortex. So here's the idea that when the rat is out there exploring the world, different areas, the hippocampus is at the top of a big hierarchy. All the sensory hierarchies end in the hippocampus. And so it's really a convergence zone for your entire sensorium. But each of these modules from the different sensory systems that projects broadly into the hippocampus will activate those hippocampal neurons during the theta, when the animal is exploring. And it's known that this is a time when there's potentiation occurring in the hippocampus. So some trace of that experience has actually been embedded into the hippocampus. Now, when you fall asleep, you go through this sequence of events I've already described. And in particular, it's known, and Ela actually mentioned this, that what happens is that the cortex goes into the slow wave mode, the slow wave theta-like mode, but now even lower frequency. And now the hippocampus is talking back to the cortex. And it does it with these sharp waves. These are very brief bursts of high frequency activity, in some cases hundreds of hertz. And this then triggers sleep spindles. So this is how the sleep spindle is initiated. And that can go on in slow wave sleep for an hour, hour and a half at the beginning of the night. But then it switches. It switches when the cortex suddenly wakes up. It gets into this REM state, and the REM now starts talking back to the hippocampus, and it cycles back and forth. And the idea is that constantly what's happening is that information is flowing from the hippocampus that was a recent experience into the cortex. And what the problem that you have in the cortex is to integrate information that might be stored in different parts of the cortex. For example, suppose that I met somebody for the first time, and I don't think we've ever met before, but was very pleased to meet you. And so I have a visual impression of you. I know how you talk, and you have a nice warm hand, right? Somatosensory, auditory, visual. That's in different parts of the cortex. And I also know your name, because it's written on your chest, right? Which is in the language area. So here's the problem. Okay, the next day, someone asks, did you see Ben Yu? And I said, oh, wait a second, yeah, I'm pretty sure she was on the program. Now I have to remember what your face looks like, and I have to remember, all the things that have happened, right? That's an event that occurred. And I want to be able to remember, long term. And so how are all those neurons connected so I can do recall? So how I can take a little piece of information and complete the pattern in different parts of the cortex. So the idea is that somehow we need to create connections long distance connections between those neurons. And that is why you have to do this. And you want to do it in a way that minimally disturbs the existing information. Otherwise, if I do it wrong, right, there may be somebody else's name that has gotten blocked. There's a funny story that Jeff Hinton once told me. His father was an entomologist who studied beetles. And he said Jeff's father refused to remember the names of any of his students because for every student's name that he remembered, he forgot the name of a beetle. Well, it's all about priorities, right? Okay, but here's the theory. The theory is that after enough cycles, and by the way, this isn't just, it turns out that you need to have the hippocampus around for weeks and months, because you're always going back and forth. For people who have lesions of the hippocampus in the region around it, they can't store new memories. In other words, they have an experience one day, they come back the next day, and they have no memory. They can be introduced to you and so forth, but they have no conscious recall of the fact that they saw you yesterday. There's some funny anecdotes along those lines that, if you tell them a bad joke, they'll laugh at it, and you come back the next day, and they'll laugh at it in exactly the same way. Old jokes don't get old if you have amnesia. Oh, okay, it's clearly, this computer has amnesia problems here. Okay, so the idea though is after enough of these cycles, after enough days and weeks and so forth, at the end, you start, you've created connections between the neurons in the different modules. And now you can get down, you restore the capacity, because the hippocampus is actually very small compared to the capacity of the cortex. So this is only a temporary. It's not even storing information. It's actually storing the links in the cortex that have to be recovered. So it's a very interesting, more like hash code if you want to use a computer science analogy. Okay, so let's now I'm going to give you one experiment, which to convince you that these sleep spindles actually might be important for memory consolidation. And this was an experiment done by Sarah Mednick. And what she does is on the first day, she gives the subject a placebo. And then they memorize a bunch of paired associates, just two random words, a bunch of them. And you go and you go to sleep, and you measure the number of sleep spindles, the density. And then when they wake up, you test to see how many you remember. So here we are. They could remember between 3 and 4. Now, the next night, what you do is you, instead of giving a placebo, or you can actually, balance it, you give them zolpidem, which is Ambien. It's a sleeping pill, interestingly. And it's known that zolpidem actually increases the density of sleep spindles, so doubles the sleep spindles. And you come back in the next morning, and you test how many they remember, and lo and behold, it's almost twice as high. Right? So this is interesting. It means that this drug, which is an intervention, you're manipulating the system and you're manipulating the spindles, really did help with remembering. Now, those of you who have used Ambien should be a little skeptical right now, because people who take it, typically, they are going on an airplane, they take it so they can sleep on the airplane. Often they end up in their hotel room, not remembering how they got there. Has anybody experienced this in their travels? No? Nobody uses Ambien?
Remember.
You don't remember? Okay. Okay, it's called a hypnotic. Okay. And so this has been shown, you know, this is a real effect. But here's the difference. The difference is that what's important here is that you do the learning first and then you take the Ambien. And it strengthens the memory. But if you take the Ambien and then do something, you can't remember it, right? So it's juggling, you know, Peter has to pay Paul, right? You know, you're boosting memory of recent past, but at the expense of the near future. It's a very interesting drug. Okay, so now comes the reveal. This is really the centerpiece of the talk, and it's something after 20 years of research I could have never expected from the animal literature. And this is a collaboration with Sid Cash at MGH, who works with epilepsy patients who come in with drug-resistant epilepsy. And the last chance they have for getting rid of it is to find out where in the cortex it's starting, right, the focus, and take it out without taking out anything that's important, like the motor system or the language system, right? That's the hope. And it turns out the temporal lobe is a hot spot where a lot of epilepsy starts, including the hippocampus, by the way. Okay, so here's an analysis that was done by Lyle Muller. And I just have to say at the onset that the traditional techniques that are used to analyze these data is basically to average over many cycles of the spindle because the signals are very weak. And when you do that, everything I'm going to tell you washes out. You have to look at single oscillations, single cycles of the oscillation, single trials, in a sense, of what is the dynamic pattern in the cortex. We've normalized the data. These are an array of 64 electrodes on the surface of the cortex called electrocorticography, ECOG. And each one of them has is recording the electrical field, and we've normalized it between 0 and 1, and then gray leveled it so that white represents the peak, and -1 represents the trough across the whole array. And so if you just focus on the one with the red dot, here's the actual oscillation, okay? And I'm going to play for you now a movie, and I'm going to ask you to, if you see any pattern in the movie that is reminiscent of something that you've seen in the movies. So just follow the peak here. This is one around. So that's about 5 cycles, 100 milliseconds per cycle. Did everybody see what I saw? It went from the temporal lobe to the parietal lobe to the prefrontal lobe. What does that remind you of? Princess Leia. Okay, it's a traveling wave. I would have never, if you had told me this, I would have said impossible.
Again.
What? Play it again. Play it again, Sam. Okay. Yeah, so it's a brief clip.
Is it real time?
No, it's 100 milliseconds per cycle, so this is really slowed down. Here's time.
And it's sort of like what you saw in the hippocampus, except in the hippocampus.
Yeah, it's linear. It's a linear traveling wave. So, okay, so here's the bottom line. If you could now go back into the literature, so I started digging back, and one of my colleagues at UCSD, David Kleinfeld, is a biophysicist, published a paper in 1990 on traveling waves in the turtle cortex. Completely, obscure paper that nobody has ever cited. Who's interested in a turtle? And second of all, a traveling wave, maybe that's just an artifact, right? I mean, what does that mean? I mean, what significance does it have? And it turns out that if you go back in literature, everything that was once called an oscillation turns out really to be a traveling wave. gamma waves, for example, gamma oscillations that have been very popular among some people and been ignored by others, saying that this is, again, it's just an epiphenomenon. That's a traveling wave. And I'm going to show you a couple more examples. I mean, it's ubiquitous. Every single oscillation. See, the concept of oscillation, the word oscillation sort of implies a synchrony, right? And that's when you have a traveling wave, you're spreading the information out over space. It's not all concentrated at the same time. And you'll see computationally that it's much more powerful than having a simple oscillation. We'll get to that. Okay, now if you do, by the way, the analytic technique that Lyell used was taken from physics, from fluid dynamics, where they had to find flows. And when you apply that to this data set, this is a single cycle, and the arrow points in degree of increasing phase. So this is a method that depends on analyzing phase, not the peak. And you need to do that because, again, they're weak, noisy signals. But in fact, you can reproduce. I mean, you can really look at the global pattern. Of all the of cycles, there's like 5,000, 10,000 cycles during the night. About half of them are circular like this. About 70% are counterclockwise, and the other 30% are counterclockwise. Some of them are expanding. Some of them, I mean, it's a much more complex pattern, but we analyze it because we really wanted to understand something having to do with this idea of memory consolidation. So if it has something with memory consolidation, maybe it has something to do with spike time dependent plasticity, right? Because that's a mechanism we know exists in the cortex, and it depends on spike timing. Okay, well, let's imagine that there was synchronous activity over the entire cortex, so that neurons were bursting here at the same point in the cycle as they were here. So here are the two local field potentials, and here are the bursts at A and B. Well, the problem is that there are time delays, long time delays across the cortex and the white matter. These are the tracks that connect the different parts of the cortex. And say that there's a 20 millisecond delay between A and B. Well, that means that after the burst in A, the synaptic inputs occur in B 20 milliseconds later. And this is post before pre. Remember, post before pre, repeated, paired at 10 hertz is going to give you LTD. Right? The 10 hertz was important. Right? If the spindle was at 5 hertz, then there'd be no synaptic plasticity at all with this mechanism. And if you have LTD, that means that all the long distance connections would be disconnected. And that's, of course, not what you want. What you want is for the arrival of the input from A to occur at the same time as the peak, and that means there has to be a difference in when they're bursting. And so if you have a traveling wave where there's a phase difference between A and B, A will burst, 20 millisecond time delay, and now you get a burst in B at about the same time, which means you're in that millisecond plus or minus 20 range. So you can increase the strengths of some synapses and decrease others. So this is compatible, right? We don't have any proof, but it just says that this could be something of why you might want time delays to actually occur and traveling waves. Now, another interesting thing has to do with how often the same pattern is repeated. And that varies with the pattern. But if you get two of those phase diagrams I showed you that are highly correlated between 0 and 1, but if you have ones that are 0.9 or above, then the very same pattern recurs over the night hundreds of times. But if the correlation is below about 0.6, then here's the shuffle control. It's basically not statistically significant, but a significant fraction, as you can see, a few of them are picked up and repeated over and over and over again, suggesting that these are the important ones. These are the ones that you do want to remember, and you're going to really take every opportunity you can to make sure that they get embedded, driven into the synapses. That's good, but is it precise enough? I mean, how much jitter is there from one of these repetitions and the next? I mean, I said that they're highly correlated, but it really depends on the relative timing of the cycle over and over again. And so here's the similarity threshold that we had in the last slide. And as you can see, these are the envelopes of the reproducibility from trial to trial, from cycle to cycle, and it increases the spread, mid-spread, is at the 5 millisecond level for the most highly correlated patterns. And of course, that's well within the plus or minus 20 millisecond range. So we're talking here about an incredible amount of precision. I mean, this is amazing. The cortex is huge. And why did nature go out of its way to create something that is so reproducible over such a large part of the cortex with a millisecond precision, unless it was taking advantage of that? And STDP, of course, is exactly matching in terms of what's needed.
How many spikes are there in one of these bursts?
Only about on the order of 2 to 3.
2 to 3.
Yeah.
So you have spike timing dependent plasticity with triples or so.
And it's very high frequency. It's on the order of, let's say, 100 to 200 hertz. At the a burst. It's really it's a low threshold calcium spike which has sodium spikes riding on top of it. Okay. Well, we wanted so of course you'd like to know, can you model this, right? And we wanted to know just roughly, looking in the literature, what have people done? So what's been very common in literature is to take a network model with local connectivity, because that's how we think the cortex is organized, with some inhibitory neurons. And lo and behold, it's not difficult to get a traveling wave. Not too surprising, because you can get recruitment. And these are what we call dense traveling waves, because in the simulations, all the neurons are going to fire in sequence. Right? And the waves, they could be linear waves, or they could be circular waves. It depends on the initial conditions and swirling patterns. That's not a good model for this, because first of all, these waves, traveling waves, only activate a very sparse number of pyramidal cells in the cortex on every cycle. Not always the same neuron on the same cycle. They're very, very sparse. They're not dense. Traveling waves that are dense occur during epilepsy, when you have massive, the activity has a tremendous firestorm of activity, and it spreads slowly, or spreading depression. And these are extremely slow compared to these. In fact, it's been measured. The time delays have been measured, and it turns out that the time delays of these long-range connections, they about 3 meters per second, are precisely in the range I was telling you about to be able to support this traveling wave. And so we wanted to know, if you just took a bunch of coupled oscillators, this goes back to Kuromoto, a Japanese applied mathematician, and is applied to explaining how it is that fireflies become synchronized in a tree, for example. And it turns out that if you have two coupled oscillators that are coupled by phase differences, so that if there's a phase difference that one of them speeds up and one slows down, then they'll all, within a few cycles, become synchronous. And this is also true of two old clocks that have pendulums. If you put them on a shelf, they synchronize because of the weak vibrations in the shelf. It's really a beautiful model. It turns out in the entire literature, nobody has ever put time delays in the Kuromoto model. Right? What would happen if you put time delays in the Kuromoto? We did that. So here it is. This is the equation for an oscillator, a couple oscillators. We put them into an array. We couple them through their phase difference. And now this is where we did something different. We put in time delays according to the distance between the locations in the cortex and between the thalamus and the cortex. It turns out that there are different time delays there as well. So here's the layout. It's our 8x8 connectivity within the cortex and the thalamus. And here's the projection. This is now a single neuron from the cortex projecting back to the thalamus, and similar beam going up, which is a little bit more tight. And now I'm going to play the simulation so you can see what that looks like. It's of course it's kind of a crude model. It's not at all realistic, but I think it'll give you an idea. It takes a couple cycles before it settles down, but when it does, right? So it's a crude circular traveling wave. However,
What's the connectivity?
The connectivity is based on the human connectome between these different areas. We know what the connectivity between the parietal cortex and the temporal cortex and the prefrontal cortex. We just put that in. I mean, this is, there's only one parameter in the whole model, and that's the coupling strength, for the feedback. And depending on what that value was, we could tune it so that it does 70% of the time in this direction and 30% in that direction.
So a pixel here is an area? Or?
A pixel here is like the electrode on the surface of the cortex. Now this is just in one hemisphere. And often people will ask, well, what about the other hemisphere, right? What's going on there? Is it doing this, or is it doing... It's actually hard to do that. And so we couldn't do it because they only put in one array. They know what's on the right side. They put the array on the right side. And it's a very invasive operation, by the way. It's really horrible to have that happen to you.
Can I ask a question? So you mentioned that the connectivity comes from real data. So I wonder, if you remove those long-distance connections and you only have the short-range connections, would you still get traveling waves of this sort?
Yeah, but they wouldn't be at the same rate or frequency. In other words, I put it this way. I think that the time delays give you an intrinsic rate at which you can propagate a sparse pattern of activity. That's the key thing, is the sparse pattern. If you only have a sparse pattern in a locally connected, it will die out. It won't go very far, right? Because there's not enough excitatory input coming in to keep the wavefront going. Yeah.
So you say that only a few neurons in a given area are participating in these waves. Is anything known about the laminar distribution of the cells that are active versus the inactive ones?
Yeah. Okay. So, well, first of all, the thalamocorticals go to layer 4, as you know, and then from there to 2, 3, and then from there to other parts of the cortex. Interestingly, in the human, we discovered that there's another pathway called the intralaminar pathway. Again, you know about this, that projects to layer 1. And it turns out that if you put a depth electrode into the cortex, you can show that there's actually 2 different spindle systems. There's one in layer 4, and there's one in layer 1 that involves layer 1 and layer 5. So the answer is that and now, okay, let's think about this from the perspective of EEG, right? Why did people see this as kind of coherent in the EEG? It was because all these cortical areas, right, the pyramidal cells are projecting up in terms of their activity patterns. And if you just have a small fraction that are coherent, that are firing, spiking, bursting at the same time, then they're going to show up, right? Because you don't need that many. The rest of them are going to cancel out if they're firing at the same time or randomly. Their contributions will be smaller. But the issue actually has to do with whether EEG is reflecting what's happening in layer 1 or deeper. My guess is it's probably layer 1.
So this would also relate to the core matrix division.
That is the difference. Okay, so that we think the core is layer 4 and the matrix is layer 1, and that layer 1 is much more diffuse. The axons come up from the intralaminar and they spread many millimeters. And so do the dendrites of layer 5 pyramidal cells. So what's going on out there is much more diffuse compared to the focal layer core, which goes to layer 4. Okay, but so what's at the end of the day, or the night? At the end of the night, when you wake up, what's going on? We think that in addition to having strengthened the connections within each module, you've also strengthened the long-distance connections between the modules. So that means now that when you have a visual input, that can excite the subset of cells in the auditory system or the language system, which then can recover the name of the person and the experience you've had, right? Even though they're in distant parts of the cortex. That's our theory. Okay, now I've already told you now that these are epilepsy patients, and there is a concern that maybe their cortex is not normal. They've had epilepsy for years and years and years. They've taken a lot of drugs. There's a concern that, in fact, their cortex has reorganized itself, and maybe this is actually a part of their that problem. Or maybe the epilepsy is really taken over the connectivity in the cortex, right? Who knows what's going on? And so I was at a meeting that was sponsored by the Kavli Institute for Brain and Mind at UCSD, and April Benesic, who studies babies, she's at Rutgers University in Newark, was giving a talk, and I was giving a talk, and at the end of the meeting, the Kavli Institute said, look, if any good ideas have come out of this meeting, we'd be happy to fund them. When's the last time that's happened to you, right? And it turned out that April and I were just talking about, you're collecting baby EEG. She studies babies that are 6 months to 18-month old during the period when they're developing language. And the idea is to try to find some markers that would help predict whether the baby would have language disability or delayed language or delayed learning. Okay, they're tightly connected. And so she's been collecting it, and she actually found some biomarkers which are really quite interesting. It's a reduced maturity, or the delayed maturity of the gamma processing signal in the cortex, which has to do with higher attentional brain functions. Okay, but the babies sleep a lot. They sleep half the time. In fact, the amazing construction going on in the first year of life, the baby is like a sponge. It's just absorbing all the sensory information and figuring out how its arm works. It puts things in its mouth, wants to understand the world, right? And that's got to get integrated into the synapses. So it turns out there's a tremendous burst of synaptogenesis during that first year. Reaches a peak, then there's pruning, and then a very slow decline, right? So there's a process here of constructing the brain. And it's maybe not a surprise that they spend half their time sleeping, because if memory consolidation is important, then they do a lot of spindling. So we collaborated with April. She gave us some of her data, and I'm going to show you. This is I showed it for the first time in New York a few days ago, and I'm still amazed when I watch this. This is looking down now. There's about 100 electrodes here, both hemispheres. This is the front of the brain. This is the back of the brain. Right, left.
Sorry, Terry. You said it's ECOG, not the infrared.
No, this is EEG.
Oh, EEG.
Oh, by the way, this is something you probably know if you've had a baby, which is that they have very thin skulls. It's soft. And it's because their brains are still expanding. The sutures haven't closed yet. And the recording, the actual I was amazed. I just didn't know this. The EEG, the strength, is as big as the ECOG. It's as if you're practically sitting on top of the cortex. It's really amazing that you can get these high-quality EEG recordings from babies.
But that's non-invasive.
What?
It's non-invasive, right?
No. The baby doesn't complain. In fact, the baby's quite happy sleeping. I mean, believe me. It's really funny. I should get a picture of it. They have this kind of geodesic dome of electrodes over them, so they look like they're kind of snowmen or something. Okay. Look at that. I mean, it's amazing. It's a very structured, highly organized traveling wave across the hemispheres. And the hemispheres, the only connection is through the callosum. So this really has to be orchestrated in a really important way.
How are we orienting? Is this a sagittal view of the
This is looking down. This is looking down. So yeah. So
Is that real time?
No. It's 100 milliseconds roughly for one swirl.
So which way is the face?
What is?
This is the front. This is the back looking down.
Is there some consistent rate at which it reverses?
Yeah, it looks like it reverses.
This is so new. We haven't had a chance to look at this. I mean, the patterns that you're seeing are really, we haven't done the phase diagrams yet, but I think it's pretty clear that there's, you're seeing many, many cycles here. So it has something.
One baby?
Pardon?
This is one baby?
We have about 10 babies. Well, in terms of humans, we had about 8 humans, but we had like hundreds of thousands. This is one baby you're seeing right now.
For about 5 seconds?
No, you're seeing for only like 1 second. You saw probably about 10 or maybe 20 cycles from one spindle. Okay, so I have one more here, which is from the side now, and they're going to rotate so you can see a different aspect.
So this is average over several...
What?
Is it an average or...
No, they're saying actual size. I mean, it's really important. I have to emphasize this. You have everything. Look, if you have the swirling thing and you average over 20 cycles, what are you going to see? You're going to see, well, you don't see nothing. You just see a constant, right? Right. So you have to look at this cycle by cycle. Yeah.
So it's very interesting that you've shown us 2 data sets with very different scales, and then your swirl happens right in the middle of what you've looked at. So I'm wondering what would it happen if, no matter what scale I looked at? The other is in your model, you have 2 time constants. One's about twice the other. Is that a principle that's going to get you this type of swirling behavior, or how does it relate?
Okay, so yeah, we've been doing a lot of modeling, and I think the critical number, or the critical parameter, it turns out to be the axonal velocity, the speed with which axons propagate information from one cortical area to the next. And actually, it's going to be a little bit different in babies because their brain is smaller. So those are a lot of interesting issues. So let me just play this other second movie for you.
I just don't see how it can be continuous across the midline, because the midline does that. So it
Oh, I see what you mean.
yeah, it doesn't make sense. There's no
Okay. So, but the point is that they're connected at the midline, right? Somehow.
The waves seem to be continuous across the midline, although there's no contiguity in brain space across the midline.
But it's EEG.
There you go. So somehow the connectivity must be created such that it ends up integrating across the cortex.
Is there any myelination in these infants?
The long distance connections in the cortex, they're called white matter because they have a lot of myelin.
Right, but at this age?
Nothing special about
It different parts of the cortex. It's interesting. There's a wave of myelination that occurs from the back to the front, but it's also regulated. The myelin is regulated in the sense that it will, and it's known for in inferior olive, for example, where timing in the cerebellum is really important. That during development, it's tuned so that you have the right time delays, millisecond time delays. In the cortex, there's recent evidence that when you're doing motor learning, that actually the volume of the cortex changes and the white matter changes, and it's thought that might be due to more myelination occurring. But these are all things that have to be looked at carefully.
So I mean, one question is also, if you think of this data and try to do source localization, is there another explanation, which is actually not a traveling wave on the surface of the entire cortex, but some sources within the brain that could create that?
Okay, so that's another good suggestion that might help Clay, because the thalamus is really the generator, and so it's the one that is doing the projections. The feedback from the cortex to the thalamus, of course, creates the spatial coherence, but we don't have our recordings from the thalamus to try to understand what the waves look like down there. They've got to be there if you see it on the cortex.
But there's no callosum for the thalamus.
But the okay, this is another interesting thing that Mircea did. Okay, he did this experiment where, the question was, is the spatial coherence coming from connections within the cortex or coming from connections within the thalamus? The reticular nucleus of the thalamus covers the entire thalamus, and it's the feedback, it's the reciprocal inhibitory feedback within the relay cells to the reticular nucleus that causes the spindle. But it turns out that if you put a knife cut through the cortex and you are looking at two parts of the thalamus, the two parts of the cortex become synchronized, as at least they thought they were synchronized, through the thalamic input. So it's the cortex going down the thalamus synchronizing within the thalamus and then going back to the cortex.
I'm just wondering how you get the wave to go across the midline, because when you first look at it, you think it's the corpus callosum doing this. But if it has to go down through thalamus, there's no route to do that very easily.
Well, it might be a combination of the thalamus. Well, you could say the same thing. How is it that the thalamus, if the thalamus doesn't have any way to communicate, how is it that the cortex, the right and left hemisphere of the cortex, get synchronized, which is what everybody has always thought, right? I don't know the answer, but I'm saying there's got to be some answer to it, right? Okay, so this is, I really think that this is, unearthing some new principles. And the question is, what computational value do they have? Is there any effect on behavior other than what's happening during sleep? And so we teamed up with John Reynolds, a colleague at the Salk Institute who studies marmosets. They have a flat cortex. And there's a part of the cortex in the middle here called MT or V5, which has cells that are directionally selective. So they're very specialized for things that are moving in the world. And it turns out that the size of that MT is just enough to be covered by a Utah array that has a 10 by 10, 100 prongs that allows you to record across the entire visual field in the MT at the same time. And here's the experiment that we did. And we were asking a question of whether the waves if there are, first, are there waves in MT in awake behaving monkeys? Number two, do they have any impact on behavior? So the task that we had them do was to fixate, and then there was after a delay, a low contrast moving Gabor. So this is like stripes that are moving across the visual field. And their task was to detect it and move their eye to it. And as you vary the contrast, their hit rate varies. And this is the fraction of times they're correct. You can see if it's high contrast, above 3.5%, which is actually pretty low, they're almost perfect. And so we set it at 50%. So they're getting it right half the time. And so they're making a lot of misses. Okay, here's a raster plot of recording from one of those electrodes at the same time that we're showing you the local field potential. And you can see that at this point the stimulus comes and there's a burst of firing which occurs, in this case, for high contrast stimulus that can be easily detected in the spike firing. So now you take it down to 50%, and then you look at how well the animal was able to detect the target as a function of the, oops, I guess I didn't even show you this in my speed to get here. Okay, this is recordings from the array. Just like in the case of the spindles from the visual cortex, you can see that the waves are traveling across. In this case, it's around 10 hertz. There's actually two waves. There's this 10 hertz wave, and there's also a much lower frequency, 2 to 4 hertz waves, which is spontaneous. Like I showed you at the beginning, there's whenever you put up a spot, there's a stimulus-evoked wave that propagates. When you make a saccade, there's a stimulus. There's now a saccade-evoked wave that starts, and this was a stimulus-evoked wave, the 10 hertz. And when you look at the phase of the wave, when do you get more hits? It turns out that it's at one phase when the cells are depolarized, when the wave is going through, and the LFP is actually at a minimum, and the membrane potential is at a maximum. Not too surprising, because that's when you're closer to threshold. So a weak stimulus will more likely produce spikes. And indeed, that's what happens if you look at the spikes in the neurons. They occur in that phase when it's more depolarized. And if you do a phase locking analysis for the hits and the misses, there's a very highly significant signal here. We're talking about 0.001. So these waves have a huge impact on behavior, right? They're there. And under these conditions, we see that you can detect the behavioral significance. But now, I want to tell you about a paper that came out in Physirev Letters just maybe a year and a half ago, which I think raises some interesting questions that actually could be relevant. These are physicists who wanted to know something about how the path, this is a silicone drop hitting an oil bath and then bouncing. As you can see, it bounced across the surface of the oil, producing ripples that then spread out every time it bounced. And they asked the following question: if at the end of the bouncing right here, we could take a snapshot of the whole pattern, can we recover the trajectory of the inputs, the pattern of inputs? And it turns out that, yes, the whole history can be reproduced. It's like a holograph or something, right? But it's a sequence in time has been transformed into a spatial pattern, right? The history over the last, maybe it took a second to do that, bang, 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 right? So a whole second of history can be recovered. And they even went further, and they introduced a shift that allowed it to bounce all the way back, showing that there's enough information so that you can reverse it. But now let's apply it to the visual system, right? V1. Now the traditional view of how information is propagated in the hierarchy is that you flash a stimulus, it gets processed in V1, and that's sent up to V2. In other words, that moment in time is passed up to V2, and V2 passes up all the way up to the top of the hierarchy where you recognize it in infratemporal cortex, right? That actually is a pretty good description of a Hubel and Wiesel experiment, right? But in the real world, you don't get flashes like that unless you're in a disco. And normally things are moving, in the background. You move, the visual scene moves, and it's much, in terms of activity patterns in the cortex, they're much denser. And we know that, in fact, that changes the level of inhibition. It produces, you get fewer spikes out when you have surround suppression. But now let's just think about that, okay, from this perspective. Suppose it was the case that as the information is flowing in continuously, it's producing these ripples and waves in the visual cortex in terms of patterns of membrane potential, depolarization and hyperpolarization, and that they're interfering with each other, right? What it means is that you've in a sense, encoded like 100 milliseconds of time of movement and activity in the visual field into a snapshot. Now, if you took a picture of that activity, you would have information not just about what's happening right now, but what's happened in the near past. And what you're passing up to V2 isn't just no longer a snapshot, it's really a movie. It's information that's been encoded over a long, significant period of time, and it's really continuous, a continuous period. So it's a history that's moving up to the top, and so what the top of the cortex is getting is much richer than a snapshot. It's getting encoded in space. It's a much richer representation of what's out there right now. Yeah.
So just to point it out, this is what Wolfgang Maas has been on about with his liquid state machine since the early 2000s, and followed on with Herbert Jaeger. And
yeah. So I think that that's a good thing that you pointed this out. In their case, they had a randomly connected network. Yeah, but they did show that the information can circulate within a network, right, in a chaotic way. In fact, liquid state machine. So and the metaphor they use is, you drop something in a pond, a stone in a pond, and you get the ripples. And I guess what I'm taking it one step further and saying, if you have a two-dimensional sheet, then it's really like a pond. It really is. And these ripples that are going out are spatially coherent as well as temporally coherent. And this is the beginning of potentially a whole new theory of vision, which could have something to do. I mean, and by the way, this is again reflecting. If you go back in the tradition, Hubel and Wiesel tradition, right? There's no room in it for oscillations. In fact, most of the systems neuroscientists from that school of vision dismiss oscillations out of hand. I'm sorry, Clay. You may not correspond to what you believe, but I can tell you that all of my friends in systems neuroscience, they've bought into the rate code, and they've bought, the time is really irrelevant. And by the way, they were inspired by the fact that Hubel and Wiesel showed that the spike timing didn't matter, right? It was really the total number of spikes. That was what was correlated with the stimulus.
It's not just buying in, though. If I got to push back a little bit here. I mean, a workaday electrophysiologist can correlate rates to all kinds of things. So,
I'm not denying that. I'm just saying that if you only look at one neuron at a time, you're never going to see a traveling wave, except it will introduce variability in your recording. Exactly the side of depends on what traveling waves are going through, whether or not you get any spikes. You see, I mean, in other words, this may explain a lot of the variability that they see. In fact, it's something like you can only account for less than half of the variance by the sensory input of the actual signal. And if I showed you in John Reynolds recordings, you can account for like 80 or 90% of the variance if you also know what the wave is doing. Yeah.
Terry, I think there's a difference in the point that you're both making. You're talking about sleep, and the idea is to communicate information that involves a temporal sequence in sleep. In the wake state, you would expect that there would be local field potential variation, but it may not have the globally coherent structure that you're seeing in these waves. In effect, the awake state is the online algorithm that somehow transiently encoding, I think is what you're saying, this structure, so that then during sleep it gets reinforced by the interaction between the traveling wave and the Buzzacchi cycle or whatever.
And that could be in different frequency bands on different scales, spatial scales, yeah.
So getting with sleep, going back to your pictures, that was during REM sleep, those traveling waves?
No, that was stage 2 sleep, which is intermediate non-REM sleep.
Okay. How about wake? Did you do similar pictures?
Well, John Reynolds was a wake behaving monkey. and there are traveling waves there at 10 hertz, which is the same frequency band as spindles, but they're not spindles. They're just transient waves that are passing through the cortex. But look, I'm not trying to say that the average fine array doesn't carry information. It does. It just doesn't carry all of it. And the importance of understanding intrinsic patterns of activity, maybe telling us something about other functions that the cortex has, which has to do with organizing the information across the spatial extent. Communication, I gave an example of that in terms of the sleep spindles. So, I think that we have to be broad in our idea of what's going on in the cortex. Yeah.
But there are two different ways to think about this oscillatory activity. One is that it gates information flow, but doesn't carry information itself. And then the other version is that the oscillation itself is somehow encoding the information. And the latter, it just doesn't seem like there are enough bits in it to me, right? I mean, it's like, you showed us this example.
Okay. So
I can.
Okay. So there's another.
More about the actual information.
Okay. I think that we have been blinded by spikes. In other words, these spikes are so definite and so pretty that somehow we really try to attribute all of the information to the spikes. And of course, over a long distance, that's true. But if you're looking at what's happening locally, there's a tremendous amount of activity that's below the level of the threshold. And I show you from Amir and Grinwald's recordings that that's got to be there, and it's affecting the spikes at some level. It carries information. What do you mean there's not enough information? I mean, the subthreshold probably has more information.
It is a very low-dimensional phenomenon, right? It's like, a coherent traveling wave. I can just describe it by like a frequency.
Okay. No. You're thinking like a physicist. No. It takes a sinner to know a sinner, right? But no. So it turns out that that's when there's a spot of light, or you drop a ball. And that's the simplest pattern because you can look at it and see. But what I'm saying is that for a real scene in the real natural world, it's incredibly complex. It's no longer a ripple or probably not a regular traveling wave because there could be interference between different locations, and it's going to be a much more complex pattern, and that could potentially carry a lot of information. By the way,
the low speed of the axons is limiting the bandwidth of these signals. Yes, tremendously.
Okay, so that's true in terms of how far it's spreading within a layer. So in terms of the long-range horizontals, they're slow, but that's okay because they're only over a few millimeters, right? But we know that V1 does a lot of local processing, so that's not surprising. But as you go up the hierarchy, right, you're getting information from larger and larger parts of the visual field, so that you're able to now reproduce these waves now that are integrating information from across the visual field. So, I think that you're raising a really interesting constraint that we have to put into the whole thing. Yes.
That we know what we are talking about. Did you try to decode this traveling wave structure in MT? I mean, can you decode stimulus properties from that?
Only insofar as we've been able to regress it against the hits and misses, right? But this is like
what we see in the hippocampus is actually that it's distortions riding on top of the traveling wave. That decode the place information. It's not the structure of the traveling wave itself, so it's a little bit different from the picture of your physicist friends with the
look, the physics experiment is a metaphor. I'm sorry, I don't take it too seriously. It's just that there you can analyze it, you can come up with a theory, you can show that the inverse problem can be solved.
For one drop.
Okay, well now that's a challenge now. Let's see if we can generalize that. Right? No, but that's exactly the point. There's a whole new set of questions here that we need to be able to explore that we just haven't even considered before. And now I want to end by just saying that part of the problem I think we have is thinking that there's only one neural code, right? That can't be the case. Nature is smarter than we are. Right? And there's no reason why nature shouldn't actually employ several codes for different conditions. For when things are changing quickly, when things are moving slowly, when it's dark, when you have very few photons, and we know that the retina has an adaptation over a vast range of logs of intensity. And why shouldn't the cortex have similar ways of adapting to different classes of stimuli? And it's just that we were so myopic in the sense that we latch onto the first thing that is reproducible, and you run with it. But the point is, at some point, you really have to start broadening your possibilities and to explore other things that now have been observed for a long, long time, not understood, these oscillations. But now that we know they're all traveling waves, right, it really opens up a whole new spectrum here. And just to finish up, this is Lyle Muller, my postdoc. Here are my collaborators. And then, like I said, I've had over 20 years of thinking about sleep spindles. And I have to say that just this recent work in humans, it wasn't discovered in animals, right? It was the human brain that really taught us something that we didn't know before. And so thank you for your attention.
Thank you. Okay, so I guess we just continue the discussion. Maybe we just follow the flow of the questions right now, and then we can transition in a more general discussion. And technically, basically at 5 o'clock or so, the buffet is outside, and we can...
Wait, are you saying there's a technical thing outside?
No. Technically speaking, at 5 p.m. I think they are starting, so there will be then the...
Oh, we're talking about liquid refreshments.
Yes, exactly.
Okay.